started.
Hello, everyone. Thanks for joining us for the CAS seminar series on this day before the snowstorm. It gives me great pleasure to introduce Dr. Chris Dimkpa. He's our new department head in analytical chemistry. He joined in August of this year. Chris got his bachelor's from Imo State Polytechnic in Nigeria in 1998, and then a master's in plant molecular biology from Catholic University in Brussels. And PhD from Friedrich Schiller University and Max Planck Institute of Chemical Ecology in Germany in 2009. Then he moved to Utah State University, first as a postdoc and then as a research assistant. And it was about 2013 when he and I first met. In 2014, he moved to the Virtual Fertilizer Research Center, which is in Washington, which is part of a larger group called the International Fertilizer Development Center, which is based in Alabama. And he moved there in 2014. And as I said, he joined us in August of this year, and I leave it to him.
Okay, good afternoon, everyone. Welcome to my presentation. I think I'm going to skip this slide. Justin already mentioned, introduced me to you guys. I've not met most of you, but I hope once we are done with the COVID, we should be able to meet and know each other very well. This picture just shows the places I've been to: Nigeria, Belgium, Germany, Utah, Washington, DC, Connecticut, and I've also been working in East Africa. And the picture there is one we took in the Sipi Falls in Uganda in 2019. So fertilizer nutrients. As you may know, we have 14 crop nutrients, but we always have much ado about NPK. Why is that so? That's because half of today's global population is fed from nitrogen fertilizers. In 2018, 120 metric million tons of nitrogen was consumed, compared to 46 of phosphorus and 34 of potassium. It goes to say that if Erisman et al. Are right, then half of us in this virtual meeting will be starving now, because we really can't do without nitrogen. But the nitrogen story is just half of the story. There are other nutrients that have specific roles, but also complement how plants respond to N, P, and K. And these include calcium, magnesium, sulfur, copper, iron, manganese, molybdenum, zinc, boron, nickel, and chloride. So my talk today will focus on the micronutrients, but more specifically on zinc. So fertilizers they are a double-edged sword. They are both a foe and a friend, in particular nitrogen. Nitrogen is a foe. It contributes to climate change factors due to release of greenhouse gases and pollutants from nitrogen losses, including nitric oxide, nitrous oxide, and ammonia. Nitrogen contributes to aquatic pollution through leaching of nitrate and into water body that cause eutrophication. Nitrogen degrades soil and causes biotic stress susceptibility by plants. We know that if we use nitrogen intensively and also phosphorus, we have an unbalanced system whereby there is less availability of other nutrients in the soil due to mining, and that reduces the tolerance of crops to pests and diseases. Nitrogen also has an effect on soil biology. It alters the soil microbial community in terms of population, diversity, and activity. All of these are well-known phenomena. However, on the other hand, nitrogen is a friend. It does contribute to improving vegetative performance. We see better growth, we see more biomass, we see more CO2 capture, which is very good for photosynthesis, which helps with mitigating climate change. It also improves yield of crops. That's the basic use of nitrogen to improve yield, which therefore enhances our food security and improve the income for the farmer. Nitrogen is also useful for fortifying of food with nitrogen in the grain. For example, this data shows how nitrogen is taken up by wheat plant, less in the roots, less in the shoot, and more in the grain. So how do micronutrients come into play? They come in because they can deliver multiple services in one go. They can increase biomass, they can enhance grain or seed yield, they can suppress disease, they can enhance tolerance to abiotic stress, such as drought, they can increase the use efficiency of nitrogen and potassium, and they can help to fortify crops with essential nutrients, mainly zinc and iron, which are very relevant in the global human health situation. And for the effect of biomass and seed yield, there is often found to be far and over and above what NPK alone can deliver. So how much of the micronutrients do we have available to us, globally speaking? We know that all of these nutrients come from the soil, except for chloride, which comes from water, from air, and also from soil. However, most of these micronutrients they are very finite in terms of their availability. So this data here shows how much of iron, manganese, and the rest of them are available as of 2014, and then how much of them are mined or have been mined over years, beginning from 2000 to 2014, averaged over 3 years. You see that what is available to us, example for iron is 82 years, and for zinc in particular, we have less than 25 years. In fact, we have about, as at then, we had 21 years of available zinc left. This is based on USGS data, and it doesn't mean it's completely accurate. There are zinc mines all over the world that are not known yet, so I guess that those will be discovered as the known ones are exhausted. But the point here is that all these micronutrients, they have no alternatives, and they are not replaceable. Some of them can interchange in enzyme activity, like nickel and like manganese and zinc, but taken together, we can't replace them. So what do we do to beat the finiteness of them? We have to recycle, we have to reuse, or we have to try to apply at lower rates. One thing we have found is that these micronutrients are, although they are taken up conventionally by plants as ions, we now know that they can be taken up as intact particles, especially with copper, with iron, with manganese, and with nickel. The issue of zinc is still conflicting. Some studies show uptake of nano, most studies show uptake of just ions. And the implication of this is that if we see uptake of nanoparticles, it could help us to lower the application rates, because each of the enzymes we use in our body or in plants cannot use more than one zinc ion at the same time, or manganese ion at the same time, and each particle contains thousands of ions. So they release one ion, the enzyme uses that ion. So we don't have to apply a lot of these nutrients as fertilizers. So this figure shows that copper can be taken up as intact particles based on SAS analysis, and these images from Jason's farmer work shows also copper in the plant, and this shows the ions, nanoparticles in the plant. So with that, we know that other mode of uptake occur with micronutrients other than ion release. So why do we have to move from conventional nutrients to nanoscale micronutrients? Well, there are a couple reasons. One is that the nutrient use efficiency for the current micronutrients is very poor. It's about 5%. We know that greater reactivity and bioactivity of nanomaterials lend them to increasing the use efficiency of micronutrients, and greater reactivity could lower application rates. I'll give you two examples here in terms of bioactivity. So this figure shows cell culture for bacteria. You apply your nanomaterials, copper oxide, 20, 100, 200, and 500 ppm. You see, at 500 ppm, all the cells are dead. There's no growth. Compare that to bulk copper at the same rate, 500 ppm. You have nearly the same effect as a control. So there is a nano size specific effect that is independent of dose. When it comes to the fungus, we see again 500 ppm. The nanoparticles have more effect in terms of reducing the hyphal diameter compared to the zinc, the bulk particle, and compared to the ions. The amount of ion here seems to be low, 89 ppm, but that is based on the amount of release of soluble ion from the particles. So it's equivalent amount of ions in terms of how much is released from the nanomaterials at a given time. And this was done for 7 days. And you see the effect on plants. this is one of the best figures you can see on the internet to show the effect of nanomaterials on biomass. You see the control, the bulk, and the zinc oxide. Clear difference, and these are at very low rates of 10 ppm. So why do I want to talk about zinc to you today in terms of moving from conventional to nanoscale? Well, zinc is, as I showed before, is very critical. It's involved in numerous over 300 enzymatic activities in biology. Those activities we see at the enzyme or cellular level, they result in agronomic outcomes, which we see in terms of biomass or grain or seed yield. About 30 to 50% of the world's soils are zinc deficient, which means they contain less than 0.5 ppm. And a third of the world's human population is zinc deficient, especially in developing countries. Only 4% of the global zinc resources is applied in agriculture. And then, and despite that low application, we see again very low nutrient use efficiency of 5%. The data I showed previously suggests that the world zinc reserves will deplete in 25 years. So based on that, we need to use less zinc in crop production, and hopefully we can do that without any agronomic penalties. If you look at the maps here, these are the global map where zinc is deficient in the soil. There are areas of the US where you find, again, spots of zinc deficiency. And if you scale up the US data, you find here that, for example, in Connecticut, up to 20 to 34% of the soils in our state here is zinc deficient. And it's worse in states like Texas, Montana, North Dakota, Kentucky, where you have up to 60% of more of the soil samples collected and analyzed showed zinc deficiency. So it may be at the global scale, the US may look to be okay, but if you dig deep into within the country, you find spots of the country where you have very serious zinc deficiency. And what does that happen? What does that mean? That translates to the humans, because if you consume these crops that are grown in zinc deficient soil, you get a effect like you see in the picture here. So our work has shown that zinc nanoparticles have various positive outcome when you use at low doses of 6 milligrams per kg or less. And some of the highlights are that they accelerate plant development under drought stress. They promote grain yield, including under drought stress. They enhance nitrogen use efficiency, and this can be plant specific. I've seen this in soybean and sorghum, but not so much in wheat. And the nanoparticles support nutrient fortification of edible tissues, grain, seed, and leaf for human consumption. So that takes us back to what you see in consuming the crop that is zinc deficient, and the effect we see in the human body. So how do we produce nanoscale zinc from conventional sources? So we can use two approaches. We can use the top-down approach, which is basically you take your mined zinc ore and you just ball mill them several times, several iterations. You get and you clean and process. You get your powder. Or you can do the bottom-up approach, where you start from the existing zinc sulfate, and you go through the process. You dissolve. You add your alkali to precipitate, and then you characterize. Using several equipment, and then you obtain you freeze dry and you obtain your powder. So regardless of which method you use, you can end up having the same type of nanomaterials. And these are pictures of some of the ones we characterize or we produce in my former place of work and characterize for different shapes and sizes. Depending on how you play around with the pH between 9 and 12, you can get these different shapes. So what does zinc do? In these studies, we looked at sorghum, and we applied nitrogen at 100 milligrams per kg, which we consider to be low. People use up to 150, 200 milligrams per kg of nitrogen to grow crops, but here we consider this to be low. And we tested the zinc nanomaterial at 6 milligrams per kg, which is again low. We compared soil versus foliar, and we compared the nano zinc plus zinc sulfate. What you see here clearly is that with nitrogen alone, you have limited growth in a zinc deficient soil. When you add your zinc, you see huge effect, whether it be nano or zinc salt. And when it comes between the nano and the zinc salt, in terms of soil application, you see that the nano does better than the salt. But in terms of foliar, the salt seems to be doing better than the nano. But taken together, there's clear effect of the nano and the ions compared to the nitrogen and phosphorus and potassium alone at low dose. So the study by the right shows soybean grown under drought stress. Drought stress means 50% feed moisture capacity. And we added the zinc at very, very low rate of 2.8 milligrams per kg, either as nano oxide or as zinc salt. What you see here in the first box is clear effect of drought on shoot biomass, grain yield, and nitrogen accumulation. Nearly half of all of these parameters taken out by drought. If you add your nanoparticles or the zinc salt under drought, you see increases that are very significant compared to the control here, the NPK alone. And in terms of the biomass, is higher with the nanoparticle than with the salt. Other parameters seem to be the same as salt and the nanoparticles for grain yield and nitrogen accumulation. The point here is that you see nitrogen accumulation is increasing with nanoparticle addition under drought stress. So that's very critical for how we manage our nitrogen. So we looked at the life cycle enhancement of nitrogen uptake by the nanoparticles under drought. You see that across the growth, all growth stages at 69 days, at 81 days, and at 131 days under drought stress. So you have non-drought, and non-drought. Under drought stress, you see effect of the nanomaterials compared to non-nanomaterials increases nitrogen uptake across all the time period, whether it be vegetative or reproductive. So that's a clear life cycle effect of the nanomaterials on how plants take up nitrogen. So it's not just an endpoint matter. In this study, we did a nutrient omission, whereby we have in some system we have all the nanoparticles that we looked at: zinc oxide, copper oxide, and boron oxide together as a bulk, as one mixture. And we did the same for the bulk non-nanoscale copper, boron, and zinc. And in each case, both of them increased the grain yield compared to the control. However, we tested to see how, by omitting each of these nutrients one after the other, what happens to the yield. So when you take out the nano zinc, see the effect goes back to the control level. So nano zinc was critical in the effect we see in this system. That effect is not shown by the bulk and bulk zinc to the same extent as the nano zinc. So there's a clear size specific effect. And we don't see that effect with boron and with copper. Again, shows clear element specific effect. And if you come down to this data here, it's clear why this is happening. The soil we used is a soil from Texas, which shows very low amount of zinc. 60% of the soils have less than 0.5. In this case, the sample we took was having only 0.1 ppm zinc, which is very, very low compared to boron, which is normal, and copper, which is normal. So clearly, not all nutrients they're not all created equal. It depends on what is in the soil. If you test similar experiment like this with a copper deficient soil, you will see clear copper effect compared to the other nutrients. So again, using the nano omission strategy, we found the effect on above ground nitrogen uptake, whereby by adding all the nanoparticles together, boron, copper, and zinc, you find clear effect on nitrogen uptake that is nanospecific, that you don't see with the bulk material. Again, if you look at the effect of omitting each of the nutrients, you find that when you remove the nano zinc and the nano copper, you have a lower amount of nitrogen in the plant. So clearly, there's nanospecific effect when you look at the all nano and the all bulk compared to the control, and there is element specific effect when you look at the zinc and the copper. Again, it goes back to the issue of how much zinc or copper is in the soil. Just some background information. Take a gulp of water. We do know that zinc and copper are urease and nitrification inhibitors. This means that they control how nitrogen is transformed in the soil up to urea, up to the nitrate. So they regulate transformation of urea to ammonium. They regulate the transformation of ammonium to nitrate, and the zinc and copper addition, in this case, minimizes nitrogen loss as ammonia and nitrous oxide. The outcome is what you see here: increased nitrogen accumulation by the plant. So looking at some mechanisms here, this fellow Kariri et al. They did some work with conventional copper and zinc, whereby they in two soils they have the urea alone shows this amount of nitrogen flux, which means how much nitrous oxide is lost from the urea, compared to when you have urea and the copper formulated together, and zinc and copper, zinc and the urea formulated together, and zinc and copper and the urea formulated together. You see the mean from these two soils. Clearly, there's an effect of having this copper, zinc, and their combinations on the amount of nitrogen lost in the system as nitrous oxide. Nothing is known in this system how nanoparticles will respond. So this is a potential scheme that I used to apply for a grant, and it shows here clearly that the urea, when you have the pathway is like this, you have your urea, you have the urea's activity, and then you have a lot of ammonia lost. Some of them go to become ammonium. Then you have the nitrification activity. A lot of it is lost as nitrous oxide and nitric oxide, and then the rest goes as nitrate, and they are all leached into the water body, and you have less uptake. However, when you add your nanomaterials, copper or zinc, you have this release of urea is either reduced or blocked. Then most of the nitrogen goes as ammonium. You can add your nanoparticles here, or you may have some residual effect from here. Again, you see the blockage here, less nitrous oxide and less nitric acid released, and then more of the nitrate also not leached out, and then you have more increase of nitrogen uptake, which is what is potentially happening in the previous slide I showed. So another thing we looked at was the acceleration of reproductive development under drought stress. Using sorghum exposed to drought at 40% feed moisture capacity, we found that the days it took, in the absence of drought, it took 43 days to get to flag leaf. Flag leaf is the last leaf in sorghum plants before you get the grain head. So under no drought, where you have the zinc added at 3 ppm, it took 43 days. Under drought, where you have the zinc added, where you have the zinc not added, it took 59 days. Under drought, where you have the zinc added, it took 47 days. So you have about 3 days reduction in the number of days it took to get to flag leaf. You can see similar occurrence with the grain head. So with no drought and with zinc, it took 46 days to get to develop the grain head. Under drought with no zinc, it took 63 days, and under drought with zinc, it took 51 days. So zinc helps these nanoparticles help to bring down the amount of time you have to wait to get flag leaf and grain head. And these are the images from this grain head data here. So at this point in time, there was no grain head with the drought and no zinc. At this picture was the same day. You have the grain head emerging already, where you have the zinc oxide at 3 ppm under drought, but under no drought and the same amount of zinc is already fully developed. So drought does something to the plant development, but zinc oxide can help to mitigate that effect. And we think that the mechanism is based on zinc being a regulator of hormones like abscisic acid and auxins, which are involved in plant-water relations and in plant phenology. So this graph shows, again, effect of panicle, effect of zinc oxide on panicle initiation. Under drought stress of 40% feed moisture capacity, we tested the zinc, 2 types of zinc, nano zinc at 1 milligrams per kg, and the bulk zinc, which is the conventional zinc, at 2 milligrams per kg. So half showed to be as good as 1. In this case, you see, it took 140 days for the plants to develop the panicle. And this is wheat. When you have under drought, you have the control. It took 143 days, so 4 more days. When you have your zinc oxide nano, it took 138 days, so as good as the control without drought. And when you have the bulk, it took even longer. So the bulk in this case didn't show any good effect in terms of lowering the number of days it takes to see your panicle under drought stress, whereas the nanoparticles did a very good job reducing that number of days by 5 days, even when you use 50% lower rate of the nanoparticles compared to the bulk. So this slide shows the zinc dynamics in a drought-stressed soil plant system. Again, with 40% feed moisture capacity, 1 milligram of zinc as nano and 2 milligrams of zinc as bulk. What you see here in the first box is that clearly drought has an effect on grain zinc and shoot zinc. In the presence of the drought, when you add your nano and the bulk, you see clear differences. Of course, you added the zinc, so you expect to see some zinc in the shoot. But the interesting thing here is that it goes also into the grain to a larger extent than the control. This control soil has low zinc, but there is still zinc in the soil. So it's not completely blank zinc. And all of this translates to a better yield for the crop under drought with zinc added compared to the control, which in this case was reduced strongly by the drought. So how does all of this meet our human zinc needs? So if you look at this this data here, it shows effect of adding zinc under no drought and the effect of having zinc at different rates, 1, 3, and 5 ppm under drought. These are the amounts of concentrations of zinc in this system. In the non-drought, you can get up to 45 ppm of zinc in the grain. Keep that in mind in the grain. And in the absence of zinc and drought, you have only 19 ppm. And when you add the zinc at the different rates, you have almost similar levels of zinc in the grain. But when you calculate using those amounts of zinc to determine your daily zinc recommended dietary allowance for a normal human being who consumes like 192 grams of sorghum grain per serving, you can see clearly that in the normal water system here, you can get up to 59% of your zinc requirements, daily zinc requirements, but just by using fertilizer to produce sorghum under normal water conditions. If you do the same for drought without any zinc, the plant can only give you up to 25% of your RDA. When you add zinc at the different rates, you can get up to 50% of your RDA just by adding the fertilizer from the soil, and then it goes into the plant and it goes into the grain under drought stress. So this has clear implications in how we can address zinc deficiency in crops, and how that can translate to addressing it in human beings who consume crops grown under drought stress with or without zinc. The story of zinc it looks very promising. However, this data here shows that when you add zinc into the soil, unlike copper and iron and nickel and manganese, it looks like you won't be able to get the zinc back in the plant tissue as nanoparticles. So here we added this zinc ion, nano zinc, bulk zinc, and all of them showed up in the plant as zinc phosphate. So the mechanism here will be that when you add the zinc to the soil as nanoparticles, most of it dissolves into ions. Some have shown very rapid dissolution of the particles into ions, and then the plant they end up in the soil in the plant as zinc phosphate. There are too many zinc and there are too many phosphorus in the plant, which is called useless phosphorus, because we apply too much phosphorus and the plants don't need all of it. They take just what they need. The efficiency of phosphorus is 15%, so the rest of it just stays in the plant and they do nothing for the plant because they're not needed in plant biology. So this is published in Wutas et al. And also in Bindraban, how we can control the so-called useless zinc and useless phosphorus, and make what we have in terms of applications more realistic. So when you have a lot of phosphorus in the plant and then you apply your zinc, it ends up as zinc phosphate, which is shown by this image here. And we know that zinc phosphate is insoluble or has low solubility, and it's almost completely non-bioavailable to humans. So what do we do with that system where you have zinc added, you have phosphorus? Both of them are not useful for human health. So it looks like we might be able to modulate this interaction by using nanoparticles. So in this image here, So this is the amount of phosphorus in the plant. When you add the zinc as nanoparticles, the amount of phosphorus goes down, just because there's formation of zinc phosphate in the soil before it gets into the plant. So you are reducing the amount of phosphorus that goes into the plant when you have the zinc, just by forming a complex that is not readily available for plant uptake. And then that translates to the fact that you have more zinc in the soil. So look at the control here and the plus zinc, residual zinc after harvest. So again, that helps us to control the amount of phosphorus that goes into the plant, but it keeps the phosphorus in the soil, which is possible that they can be used for the next planting season to grow crops. So there are two implications here. The interaction of P and zinc, it can have dual effect on P dynamics in plant soil systems. One, it can reduce the uptake of P by the plant, which is what I showed here. It can also cause a higher residual P to be in the soil, which means there's less P leaching. The P that's not taken up is left in the soil, is not leached into water bodies. So that's actually a very good thing, even though it's counterproductive in terms of uptake. So in this graph here, we show that the grain P and zinc ratio can be modulated based on the particle size. So this is the under control levels with no zinc added. The P ratio is very high, which means there will be more likelihood of forming zinc phosphate in the plant tissue. If you have the nanoparticles added, the P to zinc ratio is lower for P, and compared to the bulk is higher than the nano. This means that by using nano, we can lower the P ratio, and that will allow more zinc to be freely available in the grain for human or animal nutrition. This data is from the grain P and zinc, not the shoot. So one of the big questions is, how can we scale up, or how can we apply nanoscale micronutrients effectively and safely at the field scale? Where we have done a lot of work in the greenhouse, in the lab, but taking this to the field is a different ballgame entirely. So in many countries of the world, in developing countries, people still apply fertilizers with their hand. So this image is from India, where you can spray your bulk fertilizer in a rice field. Because they are bulk, they will come down to the ground and sink. So that's fine. You can't do that with nano, which are very small and very light in weight. So this is a no-go area. We cannot be broadcasting nanomaterials. Even by using equipment, I don't think we can do that. But this shows clearly by hand that it's not possible to do. So what is the solution? The solution may be to composite the urea or the phosphorus with the nanomaterials to generate one single product that we can efficiently and safely apply in large fields, and that will be the so-called nano-enabled bulk fertilizers. So in that respect, I have done some work formulating urea and zinc oxide nanopowder to get a composite product that farmers can use and apply their zinc and nitrogen in one go. And to do this, you have to have a binder. In this case, we use the vegetable oil as binder. Vegetable oil is bio, it's green, it's biodegradable, it's inexpensive. So that would be a good approach to look at a binder to use for binding the zinc oxide to the urea. The different colors you see here is just to differentiate the colors, because zinc oxide is whitish, urea is also whitish. So you need to show, at least have the confidence that your zinc is binding to the urea, and that you can do that by having different colors. So you can use food color of different colors to show the different efficiencies. And what I see in that, if you coat your urea with zinc oxide, whether it be bulk or not, you can have efficiency of coating as much as 74%. So that's very good process to apply in terms of formulating your urea and zinc oxide nanoparticles. So using those products, we looked at effect on grain yield when the zinc oxide is at 1% and the bulk zinc is at 2%. So 50% more zinc from the bulk. So clearly, there's no effect on grain yield when you use less zinc from nano compared to more zinc from bulk. So you have the same effect on grain yield, the same effect on zinc in the plant, But keep in mind, you are using less from the nano, 50%. So yeah, it's savings of money, savings of not adding new chemicals into the system, and all sorts of savings you can see here. And we did the same thing under drought stress, whereby we have 40% feed moisture capacity of water. And clearly, by coating the nutrients, in this, there's again no clear effect compared to non-coated, but you are using half of the amount compared to the bulk. So clearly, we can shows here that drought really has an effect, but by using the zinc oxide nanoparticles at very low rate, you can increase both the grain yield, and at the same time, you can increase the uptake of the zinc into the plant. So a few takeaways. It's very clear that inclusion of micronutrients provide multiple benefits to plants. We saw effect in the amount of zinc in the shoot and in the grain. We saw effect on nitrogen uptake. We saw effect on biomass growth. We saw effect on grain yield. We saw effect on the amount of days it takes to develop the plant systems. And from the work that Wade has done, we have also seen effect on disease control for the micronutrients, in this case with zinc. And with zinc, it appears that they are very active when the plants are stressed by environmental factors such as drought. So if we have higher reactivity based on using nanoparticles, we can achieve less amount of nutrients. We can use less amount of nutrients as fertilizers, and without any consequence for the yield. And we can do that because nanomaterials they are so tiny, they can drift, they can suspend in air and cause hazard for the handler, for the farmer, and for non-target organisms like bees and other things in the biota. So by formulating them with urea, we might be able to reduce that environmental impact, because urea is dense enough to sink to the ground. And this is applicable for countries whose farmers still spread fertilizers by hand. And even when you use the machine to spread also, but to broadcast, the same effect is also applicable. So acknowledgements. I've worked across several institutions and several collaborators. So my work at IFDC was done in collaboration with Dr. Singh and Dr. Bindraban. I've also worked with Jason. We worked together for a long time, and with Wade also. I've worked with Torres at El Paso, and I'm currently working with Swadesh Mukusantra and his postdoc at the University of Central Florida. Funding for my work has come mostly from USDA and IFA, and for the work I did in Africa, I was funded by the USAID. Thank you.
Thanks, Chris. That was great. So, Kitty, if you want to send me questions that came in, send them to the chat box. I can ask Chris those questions. And while we're waiting for those to come in, I'll actually ask a question myself. Chris, as you know, we spend a lot of time working with the NSF Chemistry Center, and they spend a lot of time optimizing properties of the particle, shape, composition, morphology. Are those types of things, do you think, going to be important for the activity of zinc, or is it really just a function of zinc release from the particle, zinc ion release?
I think, given the fact that, to my knowledge, only one paper has shown that zinc nanoparticles can be taken up by the plant. Most of it is always due to release. I don't know how much effect that shapes can have. If we can prove that shapes can affect the release itself, perhaps if you have zinc with some contour in them, not very roundish, then there could be avenues for interaction for release. So that could be where we can look at the effect of shapes on release of zinc. But in general, zinc oxide looks like it's mostly a function of the ion release.
Okay, so let me see, got a question here. In reference to your slide showing the numbers of years left for our zinc supply and other elements, the micronutrients don't break down over time, so once they're applied, where do they go? Would recycling crop residues in place, recycling of the crop residues in place conserve zinc, or is zinc going to be locked up chemically or lost to erosion?
Various things can happen. If you have them in the biomass, you can recycle the biomass and they will provide the zinc. but that will take time because it's organic. It's organic zinc then, and it has to be a function of how the biomaterial degrades. If it doesn't degrade on time, then the zinc doesn't release on time. If the zinc is left in the soil, it will definitely be fixed with phosphorus, with oxides, with other components in the soil, which makes them more or less non-bioavailable. But if the soil is acidic, then you can be sure that there will be over time there will be continuous release of the zinc from the bound phases they have in the soil.
Okay, another question: how common is zinc deficiency in humans in Connecticut? Is that an issue we see a lot of? Do you know?
I don't know so much about the human zinc deficiency in Connecticut, but I would assume that most people go to the store, to Walmart, and they pick up zinc. There are zinc supplements in the store in the US. So zinc deficiency in humans in the US may not be a big deal as it is in many countries. But I do know that in Alabama they have. We've seen talking to doctors who came to IFDC to see how we can make a linkage between plant zinc and human zinc. They do express the fact that there's zinc deficiency in parts of Alabama. I don't know if you know about the Black Belt. It's called the Black Belt of Alabama, where there is poverty and there is soil degradation. Those areas have zinc deficiency, and I think they have been able to correlate that to some human and the levels of zinc in human beings. But for Connecticut, I'm not so sure what the situation is. I think this might be something to look at. See how we can make that connection between soil, zinc, plant zinc, and then human zinc. That would be a fantastic study to do.
Mm-hmm. So in areas of the world where there is zinc deficiency in humans because of the soil, do you see that in livestock as well that are raised in those areas? Is that a problem?
Yes, it is. There is the same effect you see in the humans, you see in the livestock. So they have those livestock, you see stunted growth, not a lot of a lot of mass weight in them compared to animals fed with grass from better soils.
Okay, next up, would soil organic matter or soil minerals absorb zinc nanoparticles?
Yes, that's what it does. Zinc those are zinc for zinc, and in fact for most micronutrients, copper also, they absorb into organic matter and then they become, depending on what is in the organic matter. If it is organic acids, citric acid, enriched organic matter, the zinc may actually be more bioavailable than if it's other components, mostly clay or whatever in the soil. I think the killer for zinc will be phosphorus. That's the main killer.
Right. Next question. In several of the slides, there appeared to be no significant differences between nano and bulk zinc, copper, or boron when they're added separately, or the differences were small. Can you comment?
Yeah, but in most of those, the zinc, the nanoparticles are used at lower rates. That's the difference. Right.
So the input amount is actually half or less than half, and you're getting equivalent effect.
Yeah.
Next up, would zinc applied as a soluble salt, zinc chloride, for example, be less available to plant roots than nanoparticle zinc due to the rapid immobilization from precipitation with soil phosphorus?
What we have seen, I've seen a lot of my studies and other studies where you have equivalent. If you have zinc sulfate, I've not seen a lot of work with zinc chloride, but let's say with zinc salts, whichever it is, this effect seems to be similar in terms of uptake, because the zinc from the sulfate is readily available. It's taken up by the plant. So if you measure the plant at a given time early, you will likely find the zinc sulfate, more zinc from the zinc sulfate than the zinc nano. But if you allow enough time for this nano to transform and be taken up by the plant, at the end, based on my own endpoints, I see no superiority from the zinc sulfate compared to the zinc nano.
Does the nanoparticle zinc have to be incorporated into the soil in close proximity with the roots, or is that not that important?
I don't think it's important. Eventually, roots will find them, but of course, when you apply fertilizer, you have the 4 principles of fertilizer application, the so-called 4 R's. So one of the R's will be that it has to be as close as possible to the plant. Otherwise, they will just leach out without the roots getting to them. So yes, it should be as close as possible to the roots than not.
How about the release of zinc from so you have your zinc-coated urea. What's the kinetics of zinc release from those particles?
It all depends on the binder you use. If you use a weak binder like water, then urea is very soluble. It dissolves maybe just a few minutes better than the control. But if you use a strong binder, people have used things like gallium and all sorts of binder that will really stick the zinc to the urea. Then it takes longer to dissolve. But it won't be a matter of months. It will be weeks, and in some cases even days. So it's not a very I think one of the ways to go will be to try to see if we can incorporate the zinc into the core of the urea itself, so that as the urea dissolves from the surface, it might take days to dissolve to get to the middle. Then the zinc can be released. But then the question will arise: at what point does the plant need the zinc, early or late? So it goes back again to the soil chemistry and what is in the soil. If the soil is lacking zinc, then you want to supply the zinc as early as possible. If the soil has some zinc that can take the plant for some time, then the zinc can come in later to supplement what the soil already has. So it's a matter of the time of soil, what is in the soil, and how you want to supply the zinc to the plant.
Mm-hmm. So I've got two related questions about phosphorus in soil. So CAS, we recommend fertilizers based on 10-10-10 NPK. Are we actually recommending too much phosphorus and locking up zinc in the soil? And then the related question is, are there phosphorus deficient soils?
Yes, if we apply too much phosphorus, we are certainly locking up the zinc in the soil, and that will be worse if the soil is zinc deficient. So the little we have, we are locking up. So one way to ameliorate that will be to add some zinc into the soil, so that perchance there will be, depending on how the dynamics play out between the phosphorus and the zinc, there will be enough zinc to be taken up by the plant. Again, the timing of the application is very, very critical. What was the second question again?
Is phosphorus deficiency in soil a big problem?
Yes, in parts of the world where they use less fertilizer. Example, they use I showed the graph. I showed the amount of fertilizer used in terms of phosphorus. Those are mostly from developed countries. From developing countries, some of them use no phosphorus at all, so their soils will be phosphorus deficient. In that case, zinc may be a useful tool to help to see if they can get the phosphorus out, because there are papers showing that at low phosphorus levels, zinc can activate enzymes like alkaline phosphatase and other enzymes involved in, like phytase enzymes involved in zinc, phosphorus dynamics. So zinc is active. Zinc is active in the enzymes for those systems, so they can help to take up more phosphorus in phosphorus deficient soils. But if the soil has a lot of phosphorus, then the zinc is useless because they're all trapped by the phosphorus.
Right. Next question: are nanoparticles affected by soil cation exchange capacity?
Well, yeah, this one is if you take the way they release, if you take the release kinetics for say zinc oxide, I would say yes. I've seen in my work that potassium, when you have salts in the system, they tend to so the particles have what they call EDL, electrical. I forgot the meaning of that. It's a system that keeps the particle apart, and they don't come together. If you add salts, the salts bring that space closer. You have the particles coming together, and then they can form aggregates and lose their nano nature or their nano nature. So yes, salts do affect how nanoparticles interact with each other in terms of forming aggregates. And we know that once they form aggregates, there's less chance to solubilize and release the ions compared to the stable materials. I've seen that with calcium, with sodium, and with potassium in my work in Utah.
Okay, last question. We're obviously starting to get a better understanding of how important the soil microbiome and the rhizosphere microbiome is to plant health. Is there any reason to be concerned about adding zinc at 3, 4, 5 ppm to soil as having a negative impact on the microbiome?
No, those are the environmentally relevant zinc doses. When you start to go to 100 ppm, 50 ppm, 500 ppm, then that can be an issue. But as far as most soils are concerned, anything 20 ppm and below is all just positive outcomes you see with that level of zinc. And one of the role of nanotechnology will be to reduce even further the amount of zinc we add, because like I showed, one enzyme in our system uses only one zinc at a time. So why just add all the zinc that we end up not being used by biology to produce the crop for us?
Okay, that is all the questions I see. So I think we'll virtually thank Chris again for a great presentation. I should mention he's actually doing double duty today, working overtime, because we had a presentation this morning quite early, 6 a.m., and as well as our noontime seminar. So thank you very much, Chris, and everyone have a good day. We'll talk to you soon. Bye.
Thank you. Thank you.